How's your holidays going on? I said that enjoy your holidays. Yes, you have to enjoy, but along with whatever we are studying here in online class, you have to revise every day. Whatever we studied at a particular day, you have to revise that day notes. Okay, because this is very helpful class for you. Okay, so today we will discuss calorimetry. Okay, thermal physics. In physics, total 5 units are there. In 11th class, mechanics, then wave and oscillation, then heat and thermodynamics. 3 units are there in 11th class and 2 units in 12th class, electrodynamics and modern physics. Extra is optics. Optics also there. We can say 3 unit in 12th class also. Okay. So in third unit of 11th class, heat and thermodynamics. In this unit, this topic is there, calorimetry. So today we will discuss calorimetry. Okay. Now in this calorimetry topic, here we will discuss about only two things, two scenario, not other. Thermal expansion, that is different thing, that we will not discuss in this calorimetry. Here we will discuss if we supply heat to the body, then two possibility. One is temperature can change, another is its phase can change. Right or not? Okay. Phase means, example, if we supply heat to ice, then ice can convert into water, means solid to liquid. Okay. If we supply heat to water, then what can happen? Water can convert into vapor or steam, you can say. Agree or not? So water, liquid, steam, vapor, gas. So here also change in phase, liquid to gas. Agree or not? And its reverse also possible. Reverse means if we cool water, cool means how a body can be cooled. If we extract heat from the body, how we can extract? You put your teacup in that surrounding. After then you drink. Why? Because in some time its temperature will decrease. Means it release its heat into the surrounding. Why? Because temperature of the tea is higher, very higher than surrounding. So this is the natural tendency of heat, that it flow from high temperature to low temperature. Some students, that one girl, this Neer, she is asking that, sir, can I crack NEET by watching this session? Please reply me. Yes, whatever you know, this question you no need to ask. Why? Because in other classes, in previous classes, I discussed NEET questions also. Whatever we studied, according to that, in particular topic, questions asked in NEET exam, I solved those questions also. So by listening this class, yes, you can crack the NEET exam also. But actually these classes will not be continued throughout the year. This classes is only until you will get admission here. When classes, offline classes will start in the college, then this online classes will stop. This online classes only to help you, so that you will not waste your time in these holidays. To engage you, these classes started. Okay. So we were discussing that heat flow from high temperature to low temperature. This is a tendency, natural tendency of heat. Okay. So we were discussing that time that If we cool the body, if we extract heat from the body, then body temperature will decrease. One possibility. Continuously we are cooling the tea. So what will happen? Tea temperature decreases. Okay. Similar like that. Continuously we are extracting heat from the water. Then water temperature decreases. Okay, now water temperature reaches where? At 0 degrees Celsius. So actually what happened? After 0 degrees Celsius, water temperature cannot decrease. In surrounding, here water is there. We are extracting heat from the water. Okay. Water temperature decreases, but after 0 degrees Celsius, water temperature further cannot decrease. What will happen then? Then this water start to freeze and this water converted into ice. Water converted into ice. Is it clear? This is known as freezing point. Understood? To water temperature can decrease till 0 degrees Celsius for normal conditions. Then water converted into ice, this is known as freezing point. So this is reverse. Reverse means we are extracting heat. And if we supply heat, then what will happen? Ice convert into water. That is known as melting point. You understood melting point and freezing point? Melting point is for solid to liquid. Freezing point is for liquid to solid. Both are equal, 0 degrees Celsius. But their definition different. You all understood? Okay, students. Very well. Great. So we discussed 1 point, 2 point we discussed that if we supply heat, then temperature can increase. 1 point, another point. Phase can change, solid to liquid, liquid to gas. Reverse, if we extract heat, then what can happen? Temperature can decrease. 1 point. Another point, phase can change. How phase will change here? Vapor can convert into liquid. Liquid can convert into solid. We understood this point. Okay. So here, this point you have to write. By supplying heat to the body, its temperature can rise or its phase can change. How phase will change? That also we understood. Solid can convert into liquid. If we supply again heat, then liquid can convert into vapor. Okay. In case of water, ice can convert into water and water can convert into vapor. Clear? Okay, this is for phase change. This is phase change. Otherwise, temperature can rise. Okay, we understood that point also. Temperature can rise. If temperature rise, then it is for same state. Means water temperature can rise. So it will be water only. Ice temperature can rise. Ice can be at minus 40 degrees Celsius. Ice can be at minus 10 degrees Celsius. So if we supply heat to minus 40 degrees Celsius ice, then what will happen? Minus 40 degrees Celsius ice temperature increases, minus 30, minus 20, minus 10, then 0. Further ice temperature can't increase. Further this ice will convert into water if we supply heat continuously. That is known as melting point. Okay. Naturally, heat flow from high temperature to low temperature. Okay. Now next point. Reverse of it means if we extract heat, then what can happen? Temperature can decrease or phase can change. If phase will change, then vapor to liquid, liquid to solid, vapor to water, water to ice. Clear? Already we understood. You have to write these points, Anna. Do not sit and do not listen. Write these points in your notes copy. Okay. So reverse point. Reverse point, if heat is extracted from the body, then temperature can decrease. 1 point. Second point, or phase can change. How phase will change? Already we understood, vapor to liquid, liquid to solid. Vapor to liquid, liquid to solid. In case of water, example, steam or vapor, if we extract heat, if we condense the vapor, then what will happen? Vapor will convert it into water in case of condensation. And if we decrease the temperature of water, then water temperature reaches 0. Further it can't decrease, further it converted into ice. Is it clear? Okay. So this point we understood. Now your question is, you are thinking, sir, okay, this is theoretical part, this thing we know. But how question will come in NEET exam? Right? Our target is what? That if question will come in NEET exam from this topic, so definitely we have to correct that question. So what type of question they will ask in NEET exam? They will not ask that what is melting point, what is boiling point. Agree or not? Numerical they will ask from this topic. So how to solve those numericals, understand. First listen that which type of numericals are possible. So listen the question. So they will ask like this, that we supply 100 joule heat. You all know unit of heat. Unit of heat is joule. This is SI unit. Joule. In South India people call it joule, but it is joule. Okay, not joule. Okay, so you know that SI unit of heat is joule, right? Okay. So suppose 100 joules we supply heat to 30 degrees Celsius water. Find final state, final condition. We have to find out after supplying this 100 joule heat to 30 degrees Celsius water, final state we have to find out. Means finally it will be water, or because if we supply heat to water, then what? Here you have to come, na? You have to keep in mind these two. Okay. So this we supply heat to water. So what is the possibility? Water temperature rises until 100 degrees Celsius. This is the possibility. If we supply heat more, then what can happen? Water can convert into vapor. So both things in our mind. Agree or not? Okay. Similarly, if they given ice and we are supplying heat to the ice, minus 30 degrees Celsius ice. So what will happen first? That minus 30 degrees Celsius ice will, its temperature will rise and it will reach until 0 degrees Celsius ice. Further, if we supply heat more, then ice will convert into water. Is it clear? So 2 points. One is temperature change, another is phase change. So if there is a change in temperature, then how much heat is supplied to change in temperature of the body by some value? How much heat we have to supply to understand this point? So one is specific, 2 points are there. One is temperature change, another is phase change. So for temperature change, we defined here specific heat capacity. Understand the point. Understand this point. I am not writing first this topic. I will explain and you will say yes, definitely sir, this topic should be, this particular quantity should be specific heat capacity. You will say, understand. Suppose this is the body. Okay. Its mass is 1 kilogram. Okay. And we supply heat. How much heat? Suppose delta Q. How much heat we supply? Delta Q. Okay. Now, its temperature earlier, before supplying heat, its temperature was T1. And after supplying heat, its temperature become T2. Okay. So change in temperature is how much? T1 to T2. So change is how much? Change is delta T. Agree or not? Earlier temperature T1, we supply heat how much? Delta Q. So body temperature increases, we know, by supplying heat body temperature increases and it become T2. How much mass? 1 kilogram. So 1 kilogram body takes, 1 kilogram body absorb delta Q heat to change its temperature by delta T from T1 to T2. Agree? Okay. Now if I increase the mass, and mass is now 2 kilogram, initial temperature of this body T1, and we want that final temperature of this body should be T2. To tell me how much heat we have to supply. How much heat we have to supply here to change the temperature of this body from T1 to T2. Mass is now 2 kilogram. Same material body it is. This body material suppose copper. So this is also copper. Okay. How much heat we have to supply? Send your response. Same delta Q we have to supply again or we have to supply more? Send your response. Double of first one. Very good. Venkata, this girl, twice of. Very good. So all are saying, sir, we have to supply heat. How much? Double. 2Q. Yes. So we have to supply heat. How much? Double of this one. Normal example. We are increasing the temperature of water. Mass is how much? Suppose its volume is 10 liter. And we increase its temperature by 10 degrees Celsius. If we are taking 20 liter water, then we have to supply double heat for same change in temperature. Agree or not? Means we can say that this heat supplied depend on mass. Agree or not? This heats, if mass doubled, so heat supply also doubled. So we can say that heat supplied, this is what? Heat supplied or heat extracted, it is directly proportional to mass of the substance, mass of the body, mass of the object. You all agree or not? You all agree or not? You will say yes sir, you only told yes it will be double. So heat supplied is proportional to mass of the object. Okay, we agree. Now second example. Suppose this body mass 1 kilogram and again same body mass 1 kilogram. Its initial temperature 10 degree Celsius, final temperature 20 degree Celsius. Initial temperature 10 degree Celsius, final temperature 30 degree Celsius. Suppose initially here we have to supply how much heat, delta Q. So here how much heat we have to supply? Same delta Q or different? So you will check that here 10 degree, final 20. So how much change? To change is how much? 20 minus 10, that is 10 degrees Celsius. Agree or not? Here change is how much? 30 minus 10, 20 degrees Celsius. Agree or not? So here 10 degree, here 20. How much change? Double, right? Mass same, but temperature change doubled. So heat supplied also double. To change the temperature by double, we have to supply double heat, then only that temperature we can reach this final temperature state. Agree or not? So here if delta Q, then here it will be twice of delta Q. You all agree or not? So we can say that this delta Q, heat supplied, is also proportional to delta T. Right or not? Delta Q is also proportional to delta T. Now if we combine both, first equation delta Q proportional to M, second equation delta Q proportional to delta T. If we combine both, then it becomes delta Q proportional to M delta T. And students, we want to remove this proportional sign. So here this proportionality constant here is S, M S delta T. What is S? To remove the proportional sign, here now equal है ना, तो proportionality constant is S, and this S we call it specific heat capacity. Specific heat capacity. Now some students will say, sir, we know this formula. Arey formula you know, that doesn't matter, but how formula is derived, you have to know that basic concept, then easily you can solve questions. Right or not? So what we call this constant, this is proportionality constant, what we call it specific heat capacity. Now you can ask, sir, why it is specific? It is specific because it depends on the nature of the material. We are hitting copper. Now we are hitting here aluminium, right? So different metal takes or absorb different heat for same change in temperature. It depends on the nature of the material. Agree or not? So that's why it is specific. For copper it is different. For aluminium it is different. For iron it is different. For water it is different. So it depends on the nature of the material. Material to material it vary. That's why for a particular material it is fixed. So we call it specific. Understood or not? So what is it? Very important point, that specific heat capacity depends on the nature of the material. Okay, very important point. Specific heat capacity depends on the nature of the material. Clear? Okay, understood. So we discuss about S. Delta T, you know, temperature change, and M, you know, mass. Okay. So here you can write M is mass of the object and delta T change in temperature. Change in temperature means final temperature minus initial temperature. Is it clear? Okay, friends. Now next point. So we discussed this. S is specific heat capacity and it depends on nature of the material. That's why it is fixed at a particular material. Material to material it vary, it changes. M we discussed, delta T we discussed. Now very important point. What? When to use this formula? So if there is a change in temperature of any object, then directly you have to apply this formula. To change the temperature, how much heat is required, that is delta Q. To decrease the temperature, how much heat is required, how much heat we have to extract, delta Q. So in the question, if they ask, there is a change in temperature, water temperature changes from 30 degree to 80, 30 to 90, to how much heat we supplied, ms delta T, delta Q. Is it clear? Water temperature decreases from 70 to 10, how much heat we have to extract, you have to apply this formula, ms delta T. So I am writing this golden point that you have to remember. If there is a change in temperature, then this formula should be used. This formula okay. Now next point, we understood about temperature change. Next is, if there is a change in phase, then how to find heat supplied or heat extracted. To understand phase change, already we discussed that when phase will be changed, we are supplying heat to the ice, ice will reach till 0 degree Celsius, further its temperature will not increase, then ice will convert into water. This is phase change. So if by supplying if by supplying heat or by extracting heat, if there is a change in state, then we have to use this formula, delta Q equal to ml, delta Q equal to ml. Only two box I used, here and here. Delta Q equal to ml. What is m? M is mass, L is latent heat. You know latent word meaning is what? Latent word meaning is hidden. It is hidden. Why? We are supplying heat to the 0 degree Celsius ice, but ice temperature is not increasing. It is converting into water. So if temperature is not increasing, generally people think, array, by supplying heat, temperature should increase, but it is not increasing. So where heat is going? What is happening due to this heat? Phase change. That's why we call it latent heat. So here no temperature term. Delta Q equal to ml. If you use that formula, ms delta T 0. By that formula, delta Q will be 0. Means that delta Q is not used in change in temperature. That supplied heat is now used in changing the state. It may be from solid to liquid or liquid to vapor or reverse. Already we discussed that. Is it clear? Okay. Intermolecular energy change, Sai Kumar, yes, very good. So this delta Q is ml, m is mass and L is latent heat. When to use this formula? If there is a change in state. Is it clear? Okay. Now this latent heat depend on how this change is occurring from solid to liquid or liquid to vapor. Okay, to understand this point, if ice convert into water or water convert into ice, okay, for both we use latent heat of fusion and we represent it by LF. Is it clear? Water convert into ice, how it will convert? By extracting heat, already we discussed. Ice convert into water, how it will convert? By supplying heat. Okay, in both case, which formula we have to use? Delta Q equal to ml, and here L is which L? This is latent heat of fusion. Is it clear? Okay. So what you have to remember? Ice to water, otherwise water to ice, है ना? Water to ice. For both case, we have to use here latent heat of fusion. Now reverse. Now different phase. Liquid to vapor or vapor to liquid. Okay. Means water convert into vapor. Otherwise, vapor convert into water. Already we discussed, if we supply heat to the 100 degree Celsius water, then what will happen? Water will convert into vapor. Okay, if normal conditions are there. If we condense the vapor, extract heat from the vapor, then vapor will convert into the water. How much heat supplied or how much heat extracted? How to find this formula? Delta Q equal to ml. Now this L here, which L we use? This L is latent heat of vaporization. Here latent heat of vaporization we use. Okay. So for this change, which latent heat we use? Latent heat of vaporization. Is it clear? And we represent it by LV. It is represented by LV, latent heat of vaporization. Is it clear? Okay. Otherwise, by this diagram also you can understand, this is water converted into vapor. Otherwise, vapor converted into water. For both case, what we have to use? Latent heat of vaporization. L vaporization. Is it clear? Okay. So in this class, what we understood today? We discussed today that by supplying heat, body temperature can increase, one point. Another point, its phase can change. Okay, solid to liquid, liquid to vapor. If we extract heat, then body temperature can decrease, otherwise its phase can change. Vapor to liquid, liquid to solid. Okay. How much heat we have to supply? How to find? So two formula we discuss. If there is a change in temperature, then you have to apply this formula by closing your eyes directly. Delta Q equal to ms delta T. And if there is a change in phase, then how to find heat? How to find that heat? By using this formula, delta Q equal to ml. Here which L to this ice to water or solid to liquid, then which L? Latent heat of fusion. Water to vapor or vapor to water, then which L? Latent heat of vaporization. Now these values in the question they will give. Is it clear? Okay. So we understood today, and in next class we will discuss questions. Okay friends. So I hope that all of you enjoyed this session. Now your next class will held. Thank you so much.
In last class we have studied about basics of oxidation and reduction. That which process is oxidation, which process is reduction. So today we are going to discuss about electronic concept of oxidation and reduction. That is classical old concept. Now next is new topic is today electronic concept of oxidation and reduction. So in case of electrons, if we have to tell that any species is getting oxidized or it is getting reduced. So first point, if we write for oxidation. Now there are two possible things for any element or any atom. Either it can lose electron or it can gain electron. So if any element is losing electron, then it is called as which process? Oxidation. So what we call that oxidation is loss of electron. So what we write? Oxidation is termed as loss of electron is called as which process? Oxidation process. Now how to write that how many electrons are losing by any element? So what is reactions for that? We'll write many examples. So let us start with sodium. So if sodium is converted to sodium plus 1, Na positive, Now cation is always formed by loss of electron. So sodium is neutral. In Na plus, how much charge? Plus 1. It means how many electrons it will lose? 1 electron. So in oxidation, on which side we have to write electrons? Electrons are always written on which side? Product side. That sodium is losing 1 electron and gets converted to which ion? Na positive. Now if we take next example, if I write magnesium is converted to Mg plus 2, then how many electrons are involved? Mg to Mg plus 2. Here charge is 0. Here charge is how much? Plus 2. So how many electrons we have to write? 2 electrons on which side? Product side. Okay, so what is important here? That charge should be balanced on both sides. If you check here, magnesium 0 plus 2 minus 2. Charge on electron is taken as minus 1. So 2 electron minus 2 plus 2 minus 2, net charge is 0. So on both sides, what is charge? 0 is charge. Next, if I write Al is converted to Al plus 3. How many electrons will be involved here? 3 electron. Al 0, here it is plus 3. So plus 3 minus 3, net charge will be 0. So how many electrons are involved in Al to Al plus 3? 3 electrons are involved here. Now next, if I write example, Fe plus 2 is converted to Fe plus 3. Now this is oxidation or reduction. You have to identify where electron we have to write, on reactant side or on product side, and how many electrons we have to write. How many electrons we have to write? Fe plus 2 to Fe plus 3. Now just remember, we have to balance the charge. On reactant side, what is charge? Plus 2. On product side, what is charge? Plus 3. So can we add 1 electron here? So plus 3 minus 1. What is total charge? Plus 2. So it means it is loss of electron. And how many electron it is losing? 1 electron. So remember this, Fe plus 2 to Fe plus 3 is oxidation, because it is losing how many electron? 1 electron. Next example, if we take Sn plus 2 to Sn plus 4, it is 10. Sn plus 2 is called as stannous and Sn plus 4 is called as stannic. Now how many electrons will be involved here? On reactant side, what is charge? Plus 2. On product side, what is charge? Plus 4. So it means how many electrons we have to add? 2 electrons. Clear? Next is very important. I will write I negative converted to I2. Now you have to identify this is oxidation or reduction and how many electrons are involved here. Try to solve this. I negative to I2. You have to tell this is oxidation or reduction. Now before balancing electron, first important point, balance number of atoms also. On reactant side, how many iodine are present? 1. On product side, how many iodine are present? 2. So first balance iodine atom. So what I have written? 2 times of I negative. And here what is present? I2. Now what is total charge here? Minus 2. And what is total charge here? 0. So it means how many electrons we have to add? 2 electrons. So on this side, what we add? 2 electrons. Are you able to understand how to add number of electrons? First we have to balance atoms. So I negative 2 we have written at I2. So minus 2, here it is 0. So what we have added? 2 electrons are involved. Okay. Now let me write one more. I am writing H2 gets converted to H positive. Now find this is oxidation or reduction. Whenever electrons are present on product side, that is oxidation. So first try to balance this. H2 converted to H positive. How many electrons are involved? 2 electrons. Very good. So first we have to balance atom. How many hydrogen? 2. So here what we have to write? 2H positive. So it is balanced. Now what is charge here? 0. What is charge here? Plus 2. So how to make this 0? By adding how many electrons? 2 electron. So plus 2 minus 2, net charge will be 0. Clear? Any doubt in this? That how to write number of electrons involved? And whenever electrons are given on which side? Product side. It is which process? Oxidation. So can we say all these are example for which reaction? Oxidation reaction. So this is for oxidation. Now what we have to, next point is for reduction. So what is reverse of this? Here it is loss of electron. So in reduction it will be gain of electron. So if I write number 2, that is reduction. Now reduction is gain of electron is called as oxidation. Is called as reduction. So whenever electron is added to any species, that species is getting reduced. So we have to add electrons. It means electrons are present on which side? Reactant side. So example, if I write Fe plus 2 is converted to Fe. Now what is charge here? Plus 2. What is charge here? 0. So how to balance here? Plus 2, here 0. How many electrons we have to add? 2 electrons. On which side we have to add 2 electrons? On reactant side. Okay. So it is gain of electron or loss of electron? Gain. Fe plus 2 is gaining 2 electron and it is getting converted to Fe. So this process is called as reduction. Now I am writing 3 or 4 examples. What you have to write here? How many electrons are involved? Means you have to balance that equation. Okay. Al plus 3 to Al. Cl2 gets converted to Cl negative. O2 gets converted to O2 negative, oxide. Sulfur gets converted to sulfide. And next is Fe plus 3 gets converted to Fe plus 2. So in all these examples, what you have to write? How many electrons are involved and on which side? Reactant side or product side? Okay. Try to solve these. But whenever you are trying to balance electron, first you have to balance atoms also. Remember this thing, what we have to balance? Atom also. So Al plus 3 to Al plus 3, charge 0. So how many electrons we have to add? On which side? Reactant side. It means it is gain of electron. Now next, Cl2 to Cl negative. So first we have to balance chlorine. 2. Now here charge is 0. Here charge is minus 2. So how many electrons we have to add? 2 electrons. So Cl2 is gaining 2 electron and gets converted to 2 Cl negative. Okay. Now next, O2 negative. So first balance oxygen atom. 2. Now here charge is 0. Here charge is minus 4. So it means how many electrons we have to add? 4 electrons. So remember this, we have to balance atom also first, then balance charge. Here how many sulfur? 1 sulfur. So what are electrons? 2. 0 minus 2. So how many electrons we have added? 2 electrons. Now Fe plus 3 to Fe plus 2. So how many electron? 1. Plus 3 minus 1 plus 2. Here also charge is plus 2. Clear? So this is important. This third example, Cl2 to Cl negative and O2 to O2 negative. First we have to balance atom, then add electrons. And if electrons are coming on which side? Reactant side. It means all these are examples for reduction. That addition of electron or gain of electron is called as which process? Reduction process. So old concept is based on addition of hydrogen, oxygen, electronegative element, electropositive element, addition or their removal, which we have discussed in last class. Now today what we have discussed, electronic concept. Loss of electron is oxidation and gain of electron is reduction. So these are examples we have written. Now whenever we are writing this charge Fe plus 3, Fe plus 2, that is called as oxidation number. So there are two terms, one is oxidation number and one is oxidation state. So oxidation number and oxidation state. Okay. Now you have to tell me one thing. What do you think? These two terms are same or these two are different? That oxidation number and oxidation state are same thing or they are different thing? Only have to send that message that they are same or different. All students are saying different. Now it depends on case to case. In some cases oxidation state and number of a element can be same or it can be different. But actually these two are different terms. Clear? These two are different terms. So first is oxidation state. Actual significance is of oxidation state. There is no significance of oxidation number. So first we are going to discuss about oxidation state. Definitions are very important. Examples are very important. So listen carefully. So first I am writing about oxidation state. So O dot S means oxidation state. So oxidation state refers to the charge on an atom when all bonds of that atom are broken in a molecule. So listen carefully. If there is a molecule, in that molecule what will be there? Bonds are present between atoms. Now if we break all the bonds, then charge present on a specific atom will indicate its oxidation state. So listen, whenever any element is electropositive, so it will get which charge? Positive. If any element is electronegative, it will get which charge? Negative charge. So very simple example for this is, if we take HCl molecule, so between hydrogen and chlorine, what is present here? A single bond, covalent bond is present. Now if we break this bond, then which is more electronegative? Chlorine is more electronegative than hydrogen. So it means chlorine will get negative charge, hydrogen will get positive charge. So how it will break? H positive and Cl negative. So what is charge on hydrogen? +1. What is charge on chlorine? -1. So what we can say that oxidation state of chlorine in HCl. What is oxidation state here? -1. And what is oxidation state of hydrogen in HCl? That is +1. So when we break all the bonds of any atom, then electronegative will get negative charge, electropositive will get positive charge. So here only single bond is present. So how it will be broken? +1 and -1. So oxidation state of hydrogen in this compound is +1 and oxidation state of chlorine in this compound is -1. Just like any doubt in this, how to find oxidation state or what is definition of oxidation state? Like this, if you have any doubt in this HCl example, then tell me. Clear? So it is very simple. So first important point, you have to break all the bonds. The charge present on any individual atom is oxidation state. So we can write this point. Charge present on individual atom is its oxidation state. Now listen this, oxidation state is always integer. Either it is +1, +2, +3, +4, -1, -2, -3, -4. Oxidation state can never be fractional. So what is important point here? Oxidation state, if I am saying oxidation state is a integer, either it is positive or it is negative. So what important point other we have to remember, that oxidation state is either positive integer or it is negative integer. Oxidation state can never be fractional. That fractional term is oxidation number, not oxidation state. So first point we have discussed. Now what is oxidation number? So if we take any example, let us take in a molecule, same type of atoms are present 2 or 3. For example, H2S2O8. How many sulfur atoms are present? 2. So if we break all these bonds present in this molecule, each sulfur will get some charge. So that is called as its oxidation state. But if we take average of same type of atoms, average of oxidation state of same type of atom, then it will be referred to oxidation number. So if we write second statement for oxidation number, so oxidation number ON is average of oxidation state of same type of atoms present in a molecule. Okay. Now I will take a good example, H2S4O6. What is H2S4O6? Now if you check how many same sulfur atoms are present, 4 are present. Now if the structure you don't know, just I am writing structure for you. As chemistry will go in for 2 years, you will get all the structure, how to draw these. But now I am directly writing. This is structure for H2S4O6. Now if we break all the bonds of this sulfur, oxygen, sulfur, hydrogen, sulfur, oxygen, hydrogen, then there will be charge present on these sulfur atom. Now let us say on this sulfur, if charge is +5, on this sulfur let us say charge is 0, charge is 0, charge is +5. So when we discuss how to calculate oxidation state, then we will write this, how to find this. Just I am writing here, I am giving this in question, that what are oxidation state of sulfur? First sulfur +5, second sulfur 0, third sulfur 0, fourth sulfur is in +5. So if somebody asks me that what is oxidation state of sulfur in this compound, so what I write answer? +5, 0, 0 and +5. I will say 2 sulfur are present in +5 oxidation state and 2 sulfur are present in 0 oxidation state. But if someone asks me what is oxidation number of sulfur, now there are 4 sulfur and these are oxidation state. So definition for oxidation number, it is average of oxidation state of same type of atom. So if I take average of all these, +5, +0, +0, +5 divided by 4. So what I will get? +5/2. That means what I am getting? 2.5. That is a fraction. So oxidation number can be a fractional value, but oxidation state is never fractional. What is oxidation state? +5, 0, 0 and +5. So oxidation number is average of all these oxidation states. So how to take average? Sum of all these oxidation state divided by number of atom. How many sulfur are present? 4. So 5 + 5, 10. 10/4, how much it is? 5/2. Now check this example. Any doubt in this definition, that what is oxidation state, what is oxidation number? In next class, we will discuss that how to find oxidation state. Now this is given in question. What is given in question? That in NH2S4O6 there are 4 sulfur. When you break all the bonds, 2 sulfur is getting +5 and 2 sulfur are getting 0. 2 sulfur +5, 2 sulfur are getting 0. So it means these are oxidation state on individual atom. So on individual atom, what is charge? +5, 0, 0, +5. This is given in this example. You do not have to find. In next 1 or 2 classes, you will be able to find how to write oxidation state. But now this is given. Now we have to only find oxidation number. So oxidation number is average of these oxidation state. So 4 +5, 0, 0, +5 divided by 4. So what is answer? +5/2. So from here what I can say, that oxidation number can be fractional. Actually, there is no significance of oxidation number. Important is oxidation state, but oxidation number is average of this oxidation state. Now, it can be same or it can be different. For in this case, oxidation state and oxidation number are same or different? Different. 2 sulfur plus 5, 2 sulfur 0, that is oxidation state, but number is what one? 5 by 2. And if I take one more example, let us take, I am telling you 2 sulfur and I am giving structure for this. OH, double bond OH. Now when these bonds are broken, I am giving in question that first sulfur is present in plus 6. Second sulfur is also present in plus 6. Now tell me, what is oxidation state? In peroxide, you are calculating, student, oxidation number, not state. That is 2 oxygen atom combined. Peroxide is O2, 2 negative. So this is given in question that both sulfur are present in plus 6. So if they ask what is oxidation state of sulfur, what is answer? Plus 6 and plus 6. And if there's what is oxidation number of sulfur? Average plus 6 divided by 2, what you get? Plus 6. So in this case, number and state are same or different? In this, oxidation number and oxidation state are same. Okay. So do not focus on this, how this plus 6 is coming. This will come in next to next class, 1 or 2 classes, that how to find this plus 6 oxidation state. Now it is given in question that both are present in plus 6. So if oxidation state of both sulfur are plus 6, then what is oxidation number? Also plus 6. So in first example, oxidation number and oxidation state are different. Here they are same. So we can say that oxidation number and state can be same in some compounds, can be different in some compounds. Clear? Any doubt in this? But do not focus on oxidation state, only this definition, state and number. So which is actual term? Oxidation state. And which is hypothetical term? What we are calculating? Oxidation number, average of oxidation state. Okay, very good. Now if I take simple example, H2SO4. How many sulfur atom are present? How many sulfur atom are present? Only one. If there is only one atom, then there is no need of average. We can say oxidation number and oxidation state are same. So in this case, if I tell that oxidation state of sulfur is how much? Plus 6. Then what is oxidation number of sulfur? It is also plus 6. In H2SO4, if I am telling that in H2SO4 sulfur is present in plus 6 oxidation state. So what will be oxidation number? There is only one sulfur, so there is no role of average, so it is also plus 6. Now 3 different examples we have taken. Listen again carefully. First case is if there are more than one atoms present. If there are more than one type, same atoms present more than once. For example, H2S4O6, same sulfur is given how many times? 4 times it is given. And if their oxidation state are different, for example 2 sulfur plus 5, 2 sulfur 0, then oxidation number and state will be different. Then oxidation number and state will be different. Second example is if more than one atom are present and their oxidation state is same. For example 2 sulfur in this example and having both oxidation state, how much it is? Plus 6. So if both are having plus 6 oxidation state, then what is oxidation number? That is also same, average will become plus 6. And last case is if there is only one atom is present, means sulfur present only how much? 1. Then also oxidation number and oxidation state will be same, because there is no role of average if only one atom is present. So are you able to understand these 3 cases? that when oxidation number and state are same, when oxidation state and number are different. Now whenever we have to find oxidation number, so there are some fixed rules. For some elements oxidation number is fixed, and for some elements oxidation number can be variable. So if we start is rules to calculate oxidation number. Remember, this is rules for calculation of oxidation number, not for oxidation state. If there is only one atom, then we are finding oxidation state also. But remember, these are rules for oxidation number. After that, we will write for oxidation state. So these are very important rules. This topic is very important because from this oxidation number we can easily find out which species is undergoing oxidation, which species is undergoing reduction. So let us start from first point. If any element is present in free state, there are two states, one is free state, one is combined state. If element is combining with any other element, for example compound is formed, then it is called as combined state. If it is not combining with any other element, then it is called as which state? Free state. So if any element is in its free state or in its, we can say, elemental state, then oxidation number assigned is equal to 0. So what we write? Oxidation number for any element in its free state, or you can say elemental state, means when it is not combining with any other element, is taken to be 0. So if there is elemental state or free state, then oxidation number is taken as 0. So if I write, for example, oxygen, what is elemental state? O2. Nitrogen, what is elemental state? N2. Let us fluorine, F2, chlorine, Cl2, bromine, Br2, iodine, I2, phosphorus, P4, sulfur, S8, sodium simple, Na, aluminium, Al, mercury, Hg. So all these are free state or elemental state. Now for all these, what is oxidation state or oxidation number? Is taken to be 0. Because if same elements are present, there is no charge separation, and if only one element is present, there is no charge separation, there is no bond present. So it means oxidation number for any element in free state or in its elemental state is taken to be 0. Just check, any doubt in first point? Let's check this first point. Manjula, explain again. It is simple. If any element is in free state, it is not combining with any other element, like Na not combining with any other element, Al, Hg not combining, and one is if they are in elemental state like O2, N2, F2, Cl2, then what is their oxidation number? Oxidation number is 0. So this is about oxidation number. First point, elemental state or free state is taken to be 0. Now next is if there is any cation or anion. Oxidation number of ion is equal to charge present on that ion. Remember, not for polyatomic. Not for polyatomic. Only for single atom where charge is present. For example, if I write Fe plus 2. So what is oxidation number? Plus 2. Only single iron. What is charge here? Plus 2. If I write Al plus 3. What will be oxidation number? Plus 3. If I write Na plus. So what is oxidation number here? +1. If I write, let us take F negative. So what is oxidation number? -1. Sulfur 2 negative. What is oxidation number? -2. F negative is called as fluoride. S2 negative is called as sulfide. What is name of F negative? It is fluoride ion. S2 negative is called as which ion? Sulfide ion. Okay. So charge present on any ion, that is simple ion, not polyatomic ion, is equal to oxidation number. So Fe plus 2, oxidation number is +2, Al plus 3, +3, Na plus +1, F negative -1, S2 negative -2. So we'll continue this in next class. Only basic 2 rules we have written. So in next class we'll write 6-7 rules more, then how to find oxidation number. And today you will get that test. I have said that in last class you will get test. So from mole 20 questions will be there. So that is combined question, basics of mole concept, formulas, then some questions on stoichiometry, some questions on percentage mass, some questions on concentration term, molarity and stoichiometry. So today we'll get that in your Telegram group. So join that group and try to attempt that question. In next class I will give answer key for that 20 questions. Okay. So students, thank you for today.
Hello. Who gave scientific classification? Very good, Aristotle. Yes. Scientific attempt was first done by Aristotle, who is called as father of biology. Father of biology. Linnaeus, Aristotle, Theophrastus. Aristotle. Father of biology, Aristotle. Yes, I am fine. Aristotle. Very good. Who gave the two kingdom system of classification? Two kingdom. Two kingdom was given by Linnaeus. Five kingdom system of classification was given by five kingdoms, Whittaker. Do you remember which year he had given? 1969. Yes. What was the third kingdom that was added by Ernest Haeckel? Third kingdom. Yes, right. All of you are right. 1969. Third kingdom added by Ernest Haeckel. Plant, animals, Protista, fungi, Monera. Which one? Protista. Very good. Protista was added. Fourth kingdom added was fourth kingdom. Third one is Protista. Yes. Fourth one. Yeah, Protista. All of you are right. Fourth kingdom that was added. Monera. Yes. Correct. Monera was added. The final kingdom which was added. Final kingdom. Fifth one which was added. Fungi. Yes, Jayashree. Fungi. Very good. Very good. Which kingdoms are common in all the systems of classification? Which kingdoms are common? Yeah, fungi, okay. Yeah, my quota. Which kingdoms are common in all the 2 kingdom system, 3 kingdom system, 4, 5 kingdom system? Animalia and plants and animals. It is plants and animals. Yeah, Plantae and Animalia. They are common for all the systems of classification, all kingdoms of classification. Yes, right. Good. Now, do you remember which characters were considered by R.H. Whittaker for his classification? Is cell type considered by him or not? Cell type as a criteria for classification. Yes, very good. Very good. Yes. Then, what are the 2 cell types we have? What are the 2 cell types? Yes, correct. What are the 2 cell types we have? Mode of reproduction, yes. Mode of nutrition, right. I am asking 2 cell types are there. Yes. Harika, very good. Prokaryotic and eukaryotic cells, yes. So now you have learnt yesterday, so some 3 domains are there. Can you name the 3 domains? Name of 3 domains. Yeah, prokaryote and eukaryotic cell types, correct. 3 domains. 3 domains. Yes. Eubacteria, very good. Very good, Jahnavi, eubacteria. Then and eukarya, yeah, Deeptika. Yes, yes. Who is that? Harshit, very good, Harshit. Eubacteria, archaebacteria, or simply called as archaea, and then eukarya. These 3 are domains. Okay. So now in Whittaker 5 kingdom classification, which kingdom will be having only prokaryotes? All prokaryotes are present in that kingdom. What is that? Eubacteria, archaebacteria and eukarya. Yes, correct. Which kingdom will have all prokaryotes? Prokarya, archaebacteria, eukaryotic, prokaryotic, okay. Monera, yes. So today's our topic is Monera, first kingdom. According to the evolution. Okay. So can I say that these are the primitive organisms? Monerans are primitive organisms. So let us see here some of the primitive characteristics. Monera kingdom. Primitive organisms or first organisms. The first organisms on earth were Monerans. Okay. And this group includes all prokaryotes. This kingdom include all prokaryotes. Okay. And this all prokaryotes, Monerans, they are present almost everywhere. See, for example, they can be there in the soil, air, water, hot sulfur springs, geysers, everywhere, snow. You can't find any place without the Monerans. So, Monerans are present everywhere. You can find them, particularly the bacteria. And these Monerans, they will show all the prokaryotic features that they don't have well-defined nucleus, okay, no nucleolus, no membrane-bound cell organelles. They contain only one cell organelle, that is ribosomes. Because ribosomes are present in all, so they are called as universal cell organelles. So no membrane-bound cell organelles, no histone proteins, okay. Those are all the features of prokaryotes that will be shown by all Monerans. And in Monera, you have the examples of organisms are bacteria, which you can call them as eubacteria, then archaebacteria, then cyanobacteria, and even mycoplasmas. So there is a statement in the NCERT saying that bacteria are the sole members of the kingdom Monera. In the kingdom Monera, mainly bacteria only are there. In every term, almost you can see bacteria. So bacteria are the sole members of the kingdom Monera. The true bacteria are called eubacteria. Archaebacteria are the primitive, older than the true bacteria also, archaebacteria. Then cyanobacteria, which are also called as blue-green algae, BGA, blue-green algae. Apart from that, you have the mycoplasma bacteria, mycoplasma. Okay, these are the examples which you will find in the Monera kingdom. So what are the examples that you will find in Monera? Bacteria or cosmopolitan? Yes, correct. Cosmopolitan in distribution. Correct. But as a Monera kingdom, they are present everywhere. Okay? Yes? Okay, understood all of you? Now let us see the bacteria. First, eubacteria. Already this prokaryotic cell structure you have learnt in the cell topic only. Okay, the same thing eubacteria. These are present everywhere. As I said, cosmopolitan in nature. These are present everywhere. And this bacteria, they have simple structure. They have simple structure, but they show complex behavior. They are present everywhere. Cosmopolitan in distribution, right? So they are present everywhere. They occur everywhere. If motile forms are there, if motile, they have only flagella. They move with the help of flagella if they are motile. If they are non-motile, they don't have any locomotive structures. And this flagella, already I have told you in the cell topic that it is made up of 9+0 arrangement of microtubules. In the center, 0 microtubules will be there. In the sides, 9 triplets will be there. 9 doublets and 9 triplets I told you for centrioles and flagella. Flagella will have 9 doublets, okay? And 9+0 arrangement of microtubules will be shown by the flagella of eubacteria. They have cell wall. Cell wall is present in them, and that cell wall is made up of polysaccharides and amino acids. Polysaccharides and amino acids. Polysaccharides means sugars, carbohydrates. Amino acids, you have learnt already in biomolecules these two names, polysaccharides, complex sugars, and amino acids, the building blocks of proteins. These two combination will be forming the cell wall in eubacteria. These two combination in eubacteria will form peptidoglycan. Peptidoglycan. Peptido refers to amino acids and glycan part refers to polysaccharides. It is together called as peptidoglycan. So, cell wall in eubacteria is made up of peptidoglycan. Okay. Next one, they have different shapes. Different shapes are there. Basic structure is same, but shapes are different. About the shapes, I would like to show you the picture. You can see here, these are different shapes of bacteria. Here, first one, round ones are there. Can you see these are round ones? The round ones are called as, spherical ones or round ones are called as cocci. Spherical shaped round ones are called as cocci or coccus. Coccus is singular, cocci plural. These are rod shaped ones. Spherical ones are called cocci. Rod shaped ones are called as bacilli. Bacilli plural, bacillus singular. Then these are spring-like, spiral shape or spring-like. These are called spirillum. Spirillum singular, spirilla plural. So here the spring type will be there, spirillum. Rod shaped ones are there, bacilli. Round shaped ones are there, cocci. The one more thing which is not given will be comma shape. They will be looking like a comma. These comma shaped ones are called vibrios. Okay. If they look like a comma, punctuation mark comma, that comma shaped ones are called vibrios. So basically there are 4 shapes of bacteria. Spherical shaped ones are called cocci. Rod shaped ones are called bacilli. Spiral shaped ones are called as spirillum. Then comma shaped ones are called vibrios. So 4 different shapes are there for the bacteria. Then they show metabolic diversity. This metabolic diversity I would like to explain with the help of nutrition, mode of nutrition. So, in nutrition, these bacteria are basically of two types, autotrophic as well as heterotrophic. You know the word meaning autotrophic, I guess. Autotrophic means they will synthesize their own food materials. Heterotrophic, they depend on others for the food. So, they will show both the modes of nutrition, eubacteria. And in autotrophism, you have further two types. They are called phototrophs because they will be using the sunlight. Phototrophs will be using the sunlight and they perform the photosynthesis. Then chemotrophs will be using chemical reactions or chemical substances to get the energy to synthesize their food by photosynthesis. Both of them will do the photosynthesis, but this will use light as an energy source, this will use as chemical reactions as energy source, but all of them can synthesize their food materials. So, both the types are there in the bacteria. Then under heterotrophs you have different categories, like three categories are there, like saprophytes, which feed on dead and decaying organic matter. Then some are parasites, which will be depending on the host. Some are symbionts. They'll have mutual beneficial associationship. So under heterotrophs, they depend on others. So in that, heterotrophs, we have three categories: saprophytes, symbionts, and parasites. So all these modes of nutrition is seen in the Monera kingdom, particularly eubacteria. They will show all of that. Okay. And chemotrophs are helpful in recycling of nutrients. Recycling of nutrients. They will participate in the nutrient cycles, like nitrite, nitrogen cycle, like that they will participate here. Okay, there are various groups of organisms in the bacteria which will perform different modes of nutrition. So, as they show all the modes of nutrition, we will say that this is metabolic diversity. They are different in their metabolism, particularly in terms of nutrition. Okay. So, some bacteria are harmful because they are parasites on the host. They can cause diseases in the host, like for example typhoid. Okay, typhoid is caused by Salmonella typhi, it is a bacterium. Then cholera, Vibrio cholerae. Okay, some diseases are caused by the bacteria in humans. And not only in humans, in animals also they will cause diseases, in plants also they cause the diseases. But some bacteria are helpful for us. You know, in our intestine, colon part, one bacterium is present, E. coli, Escherichia coli. It is useful organism because it will make the vitamin B12 synthesis in the colon. From the waste material, it will synthesize the B12 vitamin. So, that is useful organism. And you must have learnt about rhizobium, heard about rhizobium. Rhizobium is a useful bacterium because it will help in the nitrogen fixation in the leguminous plants. In the leguminous plants means like which will be forming the pulses, okay, proteins like Fabaceae family. You will be learning that Fabaceae family after coming here. So, all dals, you know, no, tur dal, okay, urad dal, all dals will be having proteins. So, that dals will be having the production of that nitrogen in that plant will be happening with the help of rhizobium bacterium. Rhizobium is a particular bacterium which is a symbiont present in the leguminous plants meant for nitrogen fixation. So, that is how these different types of nutrition will be shown by the bacteria. And after that, you have the next points about the next mode of nutrition is over, mode of reproduction. So, in which way they will show the reproduction? Reproduction common in them, always, mainly it happens by binary fission. Binary fission. Binary means two. So, one bacterium, I will take for example one bacterium, rod shaped bacterium, bacilli. It will undergo binary fission. Generally, it will take 20 minutes to undergo division in the favorable conditions. and it will become 2 bacteria like this. So one bacterium, parent bacterium, divides into 2 bacterial cells by binary fission. This is the most common type of reproduction. It will come under asexual reproduction. It's a type of asexual reproduction. Okay, and this is the main type of bacterial reproduction in the favorable conditions. But in unfavorable conditions, they form endospores. They reproduce by, actually it is not reproduction, they survive the unfavorable conditions in the endospore formation. That bacterium will secrete a thick wall around it. One wall will be formed around it like this, which is many layered. Now this is called as endospore. It will develop a thick wall around each bacterial cell, and that structure is called as endospore. It will stay until all the unfavorable conditions there in their surroundings. Once the unfavorable conditions go, once the favorable conditions return, then from this endospore the single bacterial cell will be coming out. That's how they show the asexual reproduction. Both are meant for asexual reproduction. Then they also show a primitive type of sexual reproduction. They show a primitive type of sexual reproduction where from one bacterium to another bacterium the genetic material will be transferred from here. Okay, the genetic material will be transferred. So this is the DNA which is transferred from this. So in the sexual reproduction, it involves the 2 bacteria where they will be exchanging their genetic material. Particularly one bacterium sends its genetic material, DNA, to the other bacterium. This will happen in the bacteria, which is a primitive type of sexual reproduction. This is also called as genetic recombination. It is not true sexual reproduction because they are the primitive organisms. But they will be exchanging their genetic material, or transfer of DNA from one bacterium to the other, it will happen. This transfer of bacterium from one bacterium to other will be done by different methods. The one which is giving the DNA is called as donor. The one which is receiving the DNA is called recipient. So like this, they will be showing the sexual reproduction during unfavorable conditions when it is required. So this is how they will be showing the mode of nutrition, reproduction, that cell wall materials. Okay, and most of the bacteria we have learnt that plasma membrane will be showing the infoldings, nothing but mesosomes. So they will be showing one more feature after that. Next point, plasma membrane shows invaginations or infoldings. That infoldings are called as mesosomes. These are equal to mitochondria of eukaryotes because both of them will be doing the same functions. Mitochondria are involved in respiration, so these are also involved in respiration. So they are analogous, functionally similar to the mitochondria of eukaryotes. So these are the specific general characters of the eubacteria. Okay, so I hope all of you understood eubacteria. Is that clear, everyone? Yes. Any doubts? Yeah. Harshit, okay. Any doubts you have in eubacteria? No doubts? Okay, fine, Jyoti. Okay, let me proceed to the next group. you know that in Monera we have eubacteria, then archaebacteria, then cyanobacteria, and then mycoplasmas. We have just completed the eubacteria. Only one question I'll ask. Do answer for this. Later we'll proceed to the next. DNA transfer and fission occurs by same bacteria. Yeah, but not same time. Not same time. In different conditions. Okay? Yes. One question, one small question. The cell wall of, yeah, Ram Nanda, what is your doubt? You can type. What is your doubt? Cell wall of, in the meantime, cell wall of bacteria is made up of which materials? What about actinomycetes? Yes, actinomycetes are also there. Yes, filamentous bacteria, that is not given in NCERT. Okay, after coming here you will be learning extra. Very good. Thictina, polysaccharides and amino acids. Very good, very good. That is together called. What is it together called as? Polysaccharides and amino acids. I have written a word there. Yeah, N-acetyl glucosamine. Yeah. N-acetyl muramic acid, not galactosamine. N-acetyl glucosamine and muramic acid. Peptidoglycan. Very good. Peptidoglycan. Okay. What are the motile structures? Yeah, glycocalyx and all I have told you before, no? Glycocalyx, capsule, slime layer, flagella, plasmid, nucleoid. Okay, all of them. The cell topic you have to recall. Here, general characters of eubacteria. Yeah, flagella has which arrangement of microtubules? Is it 9+0 or 9+2? Only flagella. Yes, only flagella are the locomotive structures. Flagella are made up of which kind of arrangement? 9+0. Very good. So now I know that you understood. So now let us proceed to the next one, archaebacteria. Archaebacteria, which are also called as archaea. You can call them as archaea also, or archaebacteria. So these are the oldest ones, oldest monerans, because they were there when the earth was formed only. And these archaebacteria, they can tolerate extreme environmental conditions. They can survive in abnormal conditions also. Okay, like they can survive in the anaerobic conditions. Anaerobic means without oxygen they will be there. Okay, then they can survive even in the high temperatures. High temperature they can survive, high pH they can survive, high salt conditions also they can survive. Okay, because they have their cell wall material, because of cell wall material. That cell wall material which is present here, it is different and it is called as pseudomurine. Okay, and because of this different material, they can tolerate all the extreme conditions like high temperatures, high pH, high salt conditions and anaerobic conditions without oxygen. Without oxygen also they can survive, anaerobic. These are the generalized features of archaebacteria. They were the primitive oldest ones and they can stay in any conditions. Among this you have the best examples are methanogens. Methanogens, they will produce the methane gas, which is used as biogas for cooking. Methane gas is produced by the methanogens, which is a type of archaebacterium. Then these methanogens are present in the guts of ruminating animals like cows and buffaloes. Okay, which have rumen in their stomach. In them, that gut means alimentary canal, in that the methanogens are present and they are meant for cellulose digestion. In that particular part, they will help in the cellulose digestion. cellulose digestion. The methane gas in the rural areas and all, it will be used as biogas for cooking and lighting purpose. Okay. So, methanogens are very helpful because we are using that for that purpose, like biogas production, or ruminants will be using that for cellulose digestion. Then we have halophiles, which can tolerate extreme salt conditions, high salt conditions they can tolerate. Halo means salt. Then we have thermoacidophiles, which can tolerate high temperatures and high acidic conditions. Thermoacidophiles. Thermo means temperature, acido means acidic pH. So, they live in extreme environmental conditions and they can tolerate that extreme environmental conditions because of typical cell wall materials like pseudomurine. And they have in their plasma membrane also a branched chain lipids will be there. That branched chain lipids will give them the tolerance to survive in the extreme environmental conditions. These are the oldest ones. Okay, Archaebacteria, Archaea. These ones recently they found out that they show some similarities to eukaryotes, but in your textbook it is given under prokaryotes only because most of the features are similar to prokaryotes. So, it is kept under Monera kingdom. With that, Archaebacteria are completed. I hope all of you understood Archaebacteria. Yeah, any doubts are there? Yes, any doubts? Madhumita, okay. Okay. Now let us see the next one, cyanobacteria. Which are commonly called as blue-green algae. Okay. These are the first photoautotrophs. Okay. These are the first photoautotrophs on Earth to perform oxygenic photosynthesis. Oxygenic photosynthesis. That means in which photosynthesis oxygen is evolved. Okay, that photosynthesis for the first time on Earth, it was started by cyanobacteria, which are photoautotrophs. They will use the light energy to perform photosynthesis. And these cyanobacteria, they have a chlorophyll A pigment. You know which pigment is necessary for photosynthesis? That is chlorophyll. These are having chlorophyll A, which is equal to green plants. So, by having chlorophyll A, they are exactly similar to the green plants because both of them will perform the photosynthesis by the same main pigment, that is chlorophyll A pigment. Okay, that is a best part about cyanobacteria. So, they evolved, they started releasing oxygen into the atmosphere. So, first organisms to perform oxygenic photosynthesis, but photoautotrophs are there even in eubacteria also, but they were not doing oxygenic photosynthesis, they were doing anoxygenic photosynthesis, they were not liberating oxygen. These are, most of them are photoautotrophs, not most of them, all are. But here they are generally non-motile generally. Then these are unicellular ones, but they can be unicellular, they can live as filamentous forms or colonial forms. Filamentous forms means they will be looking like this. See here, it looks like a filament. One filament will be there which is made up of cells. These are all cells only, like this it will be there, filamentous forms. Colony means filaments will be there together, they will be assembled together and covered by the mucilaginous sheath. This is mucilage. and these are all filaments. So this one now it is called as colony. This is one filament. So they can be existing in the filament form, or that filaments will be enclosed within a mucilaginous sheath to form a colony. Colonial forms are also there. And this cyanobacteria, all of you can see here in this diagram, they are having normal cells. This is a normal cell, normal vegetative cell. But here you can see a big cell is there with red color thing. That is called as heterocyst. Okay, heterocyst. So in the filament you have normal cells and a big cell will be there, which is called as heterocyst. These heterocysts are meant for nitrogen fixation. So some of the cyanobacteria are useful and they will be doing the nitrogen fixation. Atmospheric nitrogen will be fixed in the soil and that will be absorbed by the plants. That nitrogen fixation doing cyanobacteria are nostoc. This is the example, nostoc and anabina. They are the nitrogen fixers. They will be fixing the atmospheric nitrogen. But some cyanobacteria are harmful and they will be forming the blooms in the water bodies, etc. Some of them are useful because they are going to fix the nitrogen, like nostoc and anabina. But if they are fixing nitrogen, they have to fix in a particular cell that is called as heterocyst. This is the cyanobacteria. Okay, I hope all of you understood cyanobacteria also, the structure. Yes. Any doubts there? Is holozoic nutrition is a part of the heterotrophism mode of nutrition? Yes, but in bacteria, in Monera, you will not see that. Holozoic mode of nutrition is not seen because you are learning here Monera. Holozoic will come under animals and protozoans. Okay. Yeah, heterocyst. Yes, Anunaya. Heterocyst is concerned with nitrogen fixation. No doubt. Okay, what are that lichens or examples of cyanobacteria? No. Lichens are not examples of cyanobacteria. Lichens are eukaryotes. It is an alga and fungal association. Symbiotic association between alga and fungal. Cyanobacteria, nostoc, anabina. Examples. Okay. The last one of today's class, mycoplasmas. In this, cell wall is absent. They don't have cell wall. Wall-less bacteria are called as mycoplasmas. Okay. These are the smallest and they live without oxygen. That means anaerobic organisms. Survive without oxygen. Okay, they don't have cell wall, so they have an outermost boundary that is nothing but plasma membrane, and they are flexible in shape because their cell wall is not there, which is rigid. So they will be present, they will be having in different shapes, no particular form or shape will be there. They are so flexible and they survive without oxygen. They don't need oxygen. Okay. Smallest cell wall less organisms which can survive without oxygen are mycoplasmas. These are the main things and they are the aerobes. First aerobes, but later they were turned into anaerobes. So they are anaerobic organisms. Okay. Is that clear? These are the only one prokaryotes or monerans without cell wall. Only one prokaryotes or monerans without cell wall. Yes, ma? Any doubts are there? Any doubts? Any doubts? Students? No doubt. Okay. Diptika. Okay, okay. No doubts. No, no. Okay. Clear. Okay. Okay. So today you have learned that Monera. So all of you by tomorrow read that Monera of NCERT new textbook if you have, or whatever I have taught you, that also you can ask questions now. Okay, very good. Tell me among these Monerans which are concerned with nitrogen fixation. Only eubacteria, only cyanobacteria, both eubacteria and cyanobacteria. Jyoti, what is it? Yeah, tomorrow Protista, yes. Tikshana, cyanobacteria. Eubacteria, all subject corner. Both. Very good, Shankar. Both. Because rhizobium is a eubacterium which is concerned with nitrogen fixation. And cyanobacteria like Nostoc, Anabina, they are concerned with nitrogen fixation. So both of them is correct. Eubacteria and cyanobacteria, right. In which cells of cyanobacteria nitrogen will be fixed? In which cells of bacteria nitrogen is fixed? A normal somatic cell, vegetative cell, or heterocyst? In cyanobacteria, which cell is concerned with nitrogen fixation? Very good. Yes, Madhuri, yeah, Kalalu, yes, yes. Heterocyst, all subject corner, yes, heterocyst. Heterocyst. Yeah. Which helps in recycling of nutrients? Photoautotrophic bacteria, chemoautotrophic bacteria, heterotrophic bacteria. Recycling of nutrients. Heterocyst, correct, yeah. Which helps in recycling of nutrients. Photoautotrophic bacteria, chemoautotrophic bacteria. Yeah, Tikshana, very good. Very good. Chemoautotrophs, yes. Good. Okay. So all of you understood now? Okay, give a thumbs up if you understood everything. Yeah. Yes. I hope all of you will read this tomorrow and come prepared so that I can ask questions. Yes. Got it? Okay, Vaishnavi, very good. Very good. Anunaya, okay, chemoautotrophic. Toxic boy, okay, very good, very good. Okay. Okay. What is that? Find my name. How can I find your name? Okay. Okay, guys. Bye. Until tomorrow.
Yes, everyone, how are you all? So you can see cockroach in your home also. Huh? So I will not be showing you cockroach. I will be teaching you about the cockroach and you can see it in your home. Okay. And or else when you will come here, you will find cockroach in the mess also. Alright. So let us start today's class. We are going to discuss about cockroach. So beta, this is given in the old NCERT. First of all, you will not find cockroach in the new NCERT. You will find cockroach in the old NCERT. But it is still there in the NEET syllabus. It is not there in the old NCERT. Okay, it is not there. It is not present in new NCERT. You will find it in old NCERT. Yes, you will find it in old NCERT. In the old NCERT and new NCERT, in both of these, there is a chapter structural organization in animals. So yes, we have talked about structural organization in animals. Yesterday itself, I have talked about animal tissues. Yes or no, everyone? So that animal tissues is also there in old NCERT. And cockroach is also there in old NCERT. So you have to study it from the old NCERT. If you don't have old NCERT, then you can just get the Xerox done for the these two topics, okay? And you can read it rest of the topics from the new NCERT. Alright. So now old NCERT, it is cockroach. Okay. Which is the unit? Structural organization in animals. This is a chapter, structural organization in animals. In that only we have studied animal tissues, and in this only we will study cockroach. Okay. So first of all, let us see cockroach belongs to which kingdom. Yes, it belongs to, is somebody kidnapped cockroach? Nobody would like to kidnap cockroach. It is not that interesting, okay? Okay, it's just that we have to study because it is the part of the NEET exam. So now, yes, which kingdom? Animalia. Which phylum of Animalia? Which phylum of Animalia? So let us see, cockroach belongs to which kingdom? Which phylum? Which class? Which order? What is the genus and what is the species? Genus and species. So you should know what is the kingdom, phylum, class, order, genus and species of the cockroach, beta. Yes. So many of you have told me the kingdom and the phylum. Can anybody tell me the class? Yes, Rama, it is Insecta. Yes, absolutely correct. So kingdom is Animalia, beta. Animalia. Cockroach is an animal. Phylum is Arthropoda. Arthropoda. This is the largest phylum of animal kingdom. Yes, you know that. Do you know that? Largest phylum of animal kingdom, Arthropoda. In this phylum, there is the largest class, beta, and that largest class is Insecta. So cockroach belongs to the largest class, Insecta. Insecta. There is another name for this class, that is Hexapoda. Hexapoda, because they have 6 legs. They have 3 pair of legs. I will tell you 3 pair of legs. Okay. What is their name? I will tell you these 3 pair of legs. So that is why they are known as Hexapoda. Then order. Order is beta Orthoptera. Orthoptera. Okay. Genus, we are going to talk about Periplaneta americana. The cockroach which we are going to talk about is Periplaneta americana. Americana. This is American cockroach. We are going to study American cockroach. Periplaneta americana. Alright. So, it has a different accent. Periplaneta americana, because it has come from America. And it is more in number than the Indian cockroach. There is the name of Indian cockroach, Blatta orientalis. So, it is more in number than Blatta orientalis, beta. Blatta orientalis. We are not going to talk about Blatta orientalis. This is Indian cockroach. Okay. We are not going to talk about Indian cockroach. We are going to talk about Periplaneta americana, because this is more in number than Blatta orientalis. Okay, Ratna, we don't talk about family in case of animal kingdom. We talk about families in case of plant kingdom mostly. So that is why here we will only talk about the kingdom, phylum, class, order, genus and species. We will not talk about the family. Okay, everyone. So are you understanding the classification? It belongs to which kingdom, which phylum, which class, which order, what is the genus and what is the species. So today we are going to talk about which cockroach. We are going to talk about Periplaneta americana. Okay, let me tell you some basic features of the cockroach. Yes, everyone, are you understanding this? Yes, understood? Yes, everyone, please give a thumbs up. Vijay Lakshmi Banot beta, because this is given in NCERT. That is the simple reason. This is given in NCERT, this is given in NEET syllabus. Okay, Periplaneta americana. You know that for every, there will be so many species, na? This is the genus. There can be so many species. We cannot study about all the species, na? So we have to choose one species from which we can, study all the features nicely. So that is why we have chosen this. Okay. And this is given in NCERT. All right, everyone, thumbs up have come. So now let us go to the next part, and that is the basic features of the cockroach. Basic features. Yes. What do you know about cockroach? You must have seen cockroach. So what do you know about cockroach? Yes. What do you know about cockroach? Beta, it runs very fast. It is a fast runner. Okay. It is a fast runner. It is a, 6 legs. So it is a fast runner, beta. Fast runner. Okay. We can call it cursorial, fast runner. Then mostly the cockroaches will come out during the night time. They are more active during the night time. Why they are more active during the night time? Because we are inactive during the night time. Now we are to sleeping, and now they will get the opportunity to go into the kitchen and to eat the leftover food into the sink, under the sink. Yes or no? On the tabletop of our kitchen, they will find something to eat. We are asleep. They are, eating in the night. So they are nocturnal. They are nocturnal, beta. Nocturnal. Nocturnal means they are active during night. They are active during night. Okay. They are nocturnal. Then, do you know that cockroach can eat anything, literally anything. It can even eat paper, wood, food. So obviously it likes food, but when they are not getting food, they are living into the bookshelves also. They are living in the cupboards also. What they are eating there? They can eat anything. So they are omnivorous. They are what? They are omnivorous. They are omnivorous. They can eat anything. Do you know that they can eat their own species also? Gee, who does that? They can eat their own species also. So they are cannibal. They are cannibal, beta. Cannibal. They can eat their own species. Their own species. If they will not get food now, they will eat their own species also. They are cannibal. So they are cursorial, they are cannibal, they are omnivorous, they are nocturnal. Where they are found? They are found in damp places. They are found in damp places. Damp places are those places where moisture is there. Moisture is there. And these places are usually you will find near to the, sink area or in the bathroom also. Okay, so in the pipes of the bathroom, they will be found because that is moisture content is there. Okay, so damp places, they are found in damp places. Then they are, beta, if you will see the cockroach, their body is shining. Okay, it seems like they have done some polishing, but they have not done any body polishing. It is simply the nature. The nature has given that polish, waxy coating, that is known as cuticle. Okay, so their body is shiny brown. Their body is shiny brown. Shiny brown. So why it is shiny, beta? Shiny. Why it is shining? Because it has waxy coating. Waxy coating of chitin. Chitin. Do you know chitin? Chitinous exoskeleton. They have chitinous exoskeleton, beta. Yes or no, everyone? You must be knowing biomolecules. Chitin. Chitin is a polysaccharide. Cuticle. Cuticle means the outermost covering. So they have a waxy coating of chitin. Okay. This waxy coating can be called as cuticle. Skin or cuticle. Our skin can also be called as cuticle. So their whole body is covered by a cuticle. And that cuticle is chitin. That cuticle is made up of chitin. Okay, chitin is a carbohydrate. It is a polysaccharide. Okay, so chitin. This chitin is, it is giving that shine to the cockroach. So that is why Rohit Deepthi is saying cute cockroach. Okay, cute. If you find it cute, okay. Okay, due to cuticle. Yes, Anunaya. So chitinous cuticle is there. They have chitinous exoskeleton. And do you know that due to the chitinous exoskeleton only they are having a successful survival on earth. That is why this arthropoda phylum is the largest phylum, because the animals of arthropoda, most of them have this chitinous cuticle or chitinous exoskeleton. Due to the chitinous exoskeleton, they are having the successful survival. That is why they are more in number. Okay, that is why this phylum arthropoda is largest in number. So chitin beta. Now I will be telling you this chitin. Interestingly, this chitin is present in form of plates. In form of plate, and this plate is known as beta sclerite. This plate is known as sclerite. So in cockroach body there are many segments. Yes, you know, arthropoda. What are the 3 phylums in which you will see segmentation? Tell me quickly. What are the 3 phylums in which you see segmentation, beta? Tell me quickly. Yes, everyone. 3 phylums in which segmentation is there. Annelida, Arthropoda and Chordata. Annelida, Arthropoda and Chordata. You will see segmentation in Annelida, Arthropoda and Chordata phylum. In us also segmentation is present. In earthworm also segmentation is present. In cockroach also segmentation is present. So they have many segments. Okay. So in every segment there are 4 sclerites. Why? Because every segment will have 4 surfaces. For example, you can see my ventral surface. Okay, now you can see my dorsal surface, and now you can see my lateral surface. So how many lateral surfaces are there? 2. 1 ventral, 1 dorsal. Okay, for example, this is the ventral surface, this is the dorsal surface, and this is the lateral surface, beta, lateral surface. So every segment will have 4 surfaces. That is why every segment will have how many sclerites? How many sclerites? Yes, everyone tell me. The sclerite is the plate of chitin. The plate of chitin is known as sclerite. And this sclerite, how many are there in each segment? 4. Okay. So, sclerites are 4 in number in each segment. In each segment. How many in number? 4 in number in each segment. These 4 are 1 sternite, 2 plurite, and beta again 1 tergite. So now you have to understand. See, look here everyone. You have to understand sternite means what? Sternite means what? Plurite means what? And tergite means what? Sternite means it is present ventrally. Ventrally. It is present ventrally. Okay, it is present ventrally. Plurite means it is present laterally. Lateral side. And tergite means it is present dorsally, beta. Dorsally. So, how many of you are understanding the meaning of sclerite, tergite, sternite, plurite? Tell me everyone. If you have not understood, please tell me. And if you have understood, please give a thumbs up. Okay? Don't talk to each other. Okay? Just get involved in the class. Yes, please tell me everyone. If you have understood, give me a thumbs up. If you have not understood, tell me what is the doubt. Okay, so most of you have understood. Yes, everyone, most of you have understood. Okay, this is, what kind of thumbs up is this, yeah? Cherry? Huh, cherry? What kind of thumbs up is this? Alright, everyone, alright. So, I repeat again, sclerite means the plates of chitin. If that plate is present on the ventral side, it can be called as sternite, because you know on the ventral side we have a bone in the chest that is known as sternum. So you can relate it to sternum, sternite. Then if it is, the plate is present on the lateral side, then it is known as pleurite. Okay, and when it is present on the dorsal side, it is known as tergite. Okay, everyone. So now moving ahead, let me tell you total how many segments are there in the body of cockroach. How many segments are there? So in the body, you will see that body is divided into 3 parts. Body is divided into 3 parts, beta. Divided into 3 parts. So what are these 3 parts? There is head, there is thorax, and there is abdomen. Head, thorax and abdomen. Head, thorax and abdomen. So if you see segments, beta, in embryo, segments in embryo, and then I will tell you segments in adult. Segments in adult. In head, how many segments in the embryo? Beta, 6 segments are there in the embryo. Okay, but all these 6 segments will fuse to make 1 segment in the head. So in the adult cockroach, in the head, there is 1 segment. Okay, 1 segment. 1 segment. In the adult, 1 segment. In the embryo's head, 6 segment. So these 6 segment will fuse to form 1 segment in the head. Then in the thorax, if you see the thorax, 3 segments are there. 3 segments are there in the thorax, and so 3 segments are there in the thorax. Here also 3 segments. If you will see the segments in the abdomen, 11 segments are there, but here you will see 10 segments. Okay, you will see how many segments? 10 segments. So total, if you will see total segments, total 6 plus 3, 9 plus 11, 20 segments are there in the embryo, whereas only 14 segments are there in the adult. Total how many segments are there in the adult? 14 segments are there. So we are going to talk about the adult only. We are going to talk about the adult. 1 head, 3 in thorax and 10 in abdomen. How many segments in head? 1. How many segments in thorax? 3. And how many segments in abdomen, beta? 10 segments in abdomen. Are you understanding this? Okay, now I'm going to tell you what are the structures present in the head, thorax and abdomen. I am going to, in today's class, we are only going to see what are the structures present in the head of the cockroach, thorax of the cockroach and abdomen of the cockroach. So let me tell you that. Let me talk about head first of all. In case of head, beta, in case of head of cockroach, there are 3 segments. There are 3 type of structures. In the head, you will find antennae. In the head, you will find antennae. Antennae will be present in the head of the cockroach. Okay. Then you will see eyes you will see. And then you will see beta mouth parts. All these things are present in our head or not? In our head, antennae are not present. In cockroach, head antennae are present. In our head, eyes are present. In our head, mouth parts are present. Upper lip, lower lip, this lower jaw, upper jaw and tongue. Okay, all these things are present, everyone. So just like that, 3 structures are present in the head: antennae, eyes and mouth parts. Antennae, eyes and mouth parts. In case of thorax, only 2 types of structures are present, beta. Only 2 types of structures are present. There are wings and there are legs. There are wings and there are legs. Wings and legs. And in case of abdomen, 4 structures you will find. One is beta anus. Next is anal cerci. Then there is anal style, and then there is genital or brood pouch, okay. So now let me tell you how many antennae. One pair of antennae, beta. How many antennae? One pair. Antennae is one pair. Okay, they help in monitoring the environment. They are sensory structures. Yes, everyone. Have you understood all the structures present in the head, thorax and abdomen? Yes, everyone. Head, thorax and abdomen. Have you understood all the structures present in head, thorax and abdomen? Beta, cloud lifestyle I will show. But once at least you should know that what are the, parts and what everything I will show. I have a picture here, I know. I have a picture here. I am going to show you the picture. But once first I will explain, then only you can understand the picture, na? If you will not listen to the notes first, if you will not listen to all these things first, then you cannot understand the picture also. Okay? So, and head, thorax and abdomen means what? Because it is there in your body also. We have a head, we have a thorax and we have an abdomen. We have a diaphragm which is separating the thoracic cavity and the abdominal cavity. So you know about all these terms. Okay. So now let us come back to the head, thorax and abdomen. How many structures in head? 3. How many antennae? 1 pair or 2. How many eyes? 2 pair. How many eyes? 2 pair. Beta, 1 pair simple eye, 1 pair simple eye, and 1 pair compound eye. 1 pair compound eye. If somebody will ask you which eye will help in the image formation, it is compound eye. It is compound eye, not simple eye. Simple eye will not help in the image formation. Okay. Then mouth parts. How many mouth parts are there? 5 mouth parts are there. 5. I will show you the mouth parts also. 5 mouth parts are there. So you should know the number. That is why specifically I am writing that. Now how many wings and how many legs? Beta, 2 pair of wings are there. 2 pair of wings are there. 2 pair. How many legs? 3 pair of legs are there. Okay, now thorax has 3 segments, right? So these 3 segments can be called as prothorax, mesothorax and beta metathorax. What are the names of 3 segments of thorax? Prothorax, mesothorax, metathorax. Okay. Prothorax is the first segment of thorax, meso is the second and meta is the third. Now these 2 pair of wings are there, na? They are connected to meso and meta. But 3 pair, there will be prothoracic leg, there will be mesothoracic leg and there will be metathoracic leg. So are you understanding? Wings are only meso and metathoracic, but legs are prothoracic, mesothoracic, metathoracic. Are you understanding wings and legs? How many? Wings total 4, 2 pair. Legs total 6 and 3 pair. Are you understanding this? Coming to the abdomen. Anus. Anus is meant for the egestion. Anus is present in, anus is single opening. It is present in 10th segment, beta. 10th segment. Which side? Dorsal side. So we can tell 10th tergum. We can tell what? 10th tergum. Here only anal cerci is present. Anal cerci is present in the 10th tergum only. It is paired, means it is 1 pair, means it is 2. Okay. So 1 pair. Similarly, anal style is also 1 pair, beta. But anal style is only present in male. Although females are more stylish, but cockroach male is more stylish. Okay, human females are more stylish, but cockroach male is more stylish. Why we are saying cockroach male is more stylish? Because cockroach male has anal style. Female does not have the structure anal style, beta. Everyone, are you understanding? Anal style is present only in male cockroach. So it is a character of sexual dimorphism. It is a character of sexual dimorphism. So anal style is present in male. It is present in males only. It is present in males only. It is one pair. It is one pair. And which segment? It is ninth segment. Ninth segment. Ninth sternum means ventral side. Sternum means ventral side. Sternum. Sternum means ventral side. Tergum means dorsal side. Okay. Okay. So now, and genital or brood pouch. Genital or brood pouch is helping in the reproduction. It is different in male, different in female. If I will tell you in male and female, genital pouch beta. In case of male, it is formed by two segments, ninth sternum plus ninth and 10th tergum beta. Ninth sternum and 9th and 10th tergum. If you see the genital pouch in case of females, it is formed by three segments, 7th, 8th and 9th sternum. That means ventral side. Whereas you will see 7th sternum is boat shaped. It is boat shaped. 7th sternum is boat shaped. Okay. So I have told you all the three parts. Head, thorax and abdomen. I have told you what are the structures present in head, thorax and abdomen, and now I am going to show you all these structures from the diagram. Yes, everyone. So are you understanding all this? So I have given you a brief introduction of the structures present in head, thorax and abdomen. Detailed, details about all these structures we will study when you will come to the class. Okay, Ananya, thank you. Crystal clear. Okay. Okay, everyone. Yes. Everyone understood? Now, are you ready to see the cockroach? So, are you able to see this figure, everyone? Are you able to see this figure, everyone? Here you can see the head. Yes or no? Here you can see the head, beta. Head. What is written? See, head is written. Okay, head is written. Then after head, this is thorax, beta. This is thorax. This is thorax. This is thorax. Prothorax can also be called as pronotum. Prothorax can also be called as pronotum. Then there is mesothorax and then there is metathorax. So these are the three parts of the thorax. And then there is abdomen. Then there is abdomen. This is abdomen, 10 segments. From here to here, this is abdomen, beta. Now you can see in the head, you can see antennae, filiform antennae. How many pair? One pair. So two are there. Okay. Then you can see compound eye. You can see compound eye. There is one more structure in NCERT where you can see simple eye also and compound eye also. Okay. So here you can see compound eye, one pair. Then you can see the wings, beta. This wing is mesothoracic wing. Mesothoracic wing. What is the another name of mesothoracic wing? Tegmina, mesothoracic wing. And what is the name of the metathoracic wing? Hind wing. Metathoracic wing or hind wing. If somebody will ask you which of this help in the flight, Which of this help in the flight? So let me tell you, the mesothoracic wing helps to cover, but the one which is broad, it helps in flight, beta. So hind wing helps in flight, okay. Why? Because it is lightweight, it is broad, that is why, okay. Then you can see abdomen, okay. In the thorax you can see wings, and in the thorax you can see legs, prothoracic leg, mesothoracic leg, metathoracic leg. All these legs are beta 5 segmented, all are same, and in all the legs there are 5 segments. In every leg, in each leg there are 5 segments, okay. And then comes to the abdomen. You can see anal cerci, you cannot see anal style here, but you can see anal cerci, abdomen. So anus is there, anal cerci is there, and anal style is there. Are you understanding this? Yes or no, everyone? Then let me show you the mouth parts also. So you can see the mouth parts also. Here if you will see, you can see the mouth parts. Yes, this diagram. In this diagram, you can see the antennae. This is the head of the cockroach. You can see simple eye. This is simple eye, beta. Simple eye. And you can see compound eye. And you can see antennae. And then you can see the mouth parts. Mandible, maxilla, labrum, labium, and hypopharynx. So here, mandible. How many mandible? 1, 2. That means mandibles are paired. How many maxilla? 1, 2. That means maxilla are also 2 in number. Paired. How many labrum? Upper lip? 1. How many labium? Lower lip? 1. How many hypopharynx? Tongue? 1. These are the 5 mouth parts. Mandible, maxillae, 1 pair. Labrum, 1. Labium, 1. And hypopharynx, 1. Okay, these are the 5 mouth parts. This is the head region. You have seen the thorax region and the abdomen region. I have told you all the structures. The name of all the structures in the head, thorax, abdomen. In the head, antennae, eyes, mouth parts. In the thorax, wings, legs. In the abdomen, anus, anal cerci, anal style, genital or brood pouch. Genital or brood pouch is the part of the reproduction. Okay. So, you can see here genital pouch. How many segments? 3 segments. 7th, 8th and 9th segment. From which side? From ventral side, sternum. So, 3 segments are involved in the formation of brood pouch of female. But only 2 segments are involved in the formation of brood pouch of males. Which are those 2 segments? 9th and 10th. 9th segment is involved from both the sides, ventral side as well as dorsal side. So 9th sternum and tergum, and 10th only on the dorsal side, 10th tergum. So 9th sternum, 9th and 10th tergum. Here 7th, 8th and 9th sternum. Here the 7th sternum, very important point written in NCERT, is boat shaped. It is in the shape of a boat. Okay, so that was all about the external morphology of the cockroach. Today we have studied the external morphology of the cockroach, the basic features of the cockroach, and the cockroach belongs to what is the classification of the cockroach. Okay, Anunya, thank you so much. So everyone, please give me a thumbs up if you have understood today's class. Yes, everyone, give me your heart if you have understood today's class. मैम पूरा समझ गई. शंकर मन्ना, थैंक यू बेटा. Oh, Lakshmi, you are also Yadav. Okay, everyone, yes, you understood? Okay, so let's meet in tomorrow's class. Okay, that was all for today's class. Don't forget to hit the like button. If you have understood, if you are getting benefit from all these classes, you should like at least, है ना everyone, you should like. Okay everyone, so see you tomorrow. I think tomorrow I am going to teach you breathing and exchange of gases chapter. Okay, thank you Rakesh. Okay, see you tomorrow everyone. Bye bye, take care, God bless you all.